He's dating up to and seeing that stuff, and I was just like sitting there
This half is always full, and this half is always empty.
I don't know.
Well, good morning, everybody. Welcome to the second lecture on transplantation. So last time we kind of went through an introduction about how transplant immunology is really just kind of a variation on the theme of the regular immunologic adaptive response. We'll kind of go into that a little further today. And we had a case, Susie Medeiros, who had a kidney transplant, and you had an opportunity to develop your own learning issues of surrounding transplant, surrounding histocompatibility, maybe surrounding complications, and then post-transplant follow-up. I don't know, I sent out a case yesterday to kind of look at that we're going to go over. I don't know if anybody kind of has a chance to look at that. But some of the objectives today are to talk about the role of histocompatibility testing for solid organ transplants, talk about the different types of transplant rejection, explain the underlying immune mechanisms behind those, talk about a little bit about the MOA of the anti-rejection drugs, and then talk about some of the complications. And really, at this point, it can really only be kind of an overview and maybe reinforcing some of the ideas you talked about when you presented your learning issues. So we showed this slide, I showed this slide before, and the question is, what causes transplant rejection? That is, what is the barrier to the ability to transplant organs without having them be rejected at some point? And so after a lot of work with mice, we discovered that there is a genetic locus that codes for a bunch of proteins that eventually was called the MHC proteins, and the locus, the genetic locus, was the MHC complex. And in humans, this worked out to be the HLA system. So for human skin grafts, the transplant antigens that cause rejection are these HLA antigens. And we talked about what they were. So we know that transplant rejection is both the cause of the foreign tissue response and also the effector of that. So the peptide epitopes on the HLA proteins act as foreign antigens, and we need the HLA polymorphism to be able to fight off a variety of pathogens. And the transplanted tissue that stimulates the immune response is called alloreactivity in the recipient. And on the effector side, the antibodies and T cells of the adaptive immune response respond to the HLA epitopes that are being presented to the recipient to reject organs. And then I showed this slide the last time, just to remind you that the MHC system in humans, the HLA system, consists of class 1, class 2. The main ones for class 1 are A, B, and C, and then the non-classical HLA class 1s are E, F, and G. I think there might even be a couple more. Now, class 2, the HLA DR, DQ, and DP, and they're found on the antigen-presenting cells, primarily dendritic cells, B cells, macrophages, and thymic cells. And it turns out we said they're the most polymorphic and polygenic system because we have developed this over time to be able to present pathogenic peptides from different pathogens to be able to kick off this adaptive immune response. And when a lot of these were discovered, we didn't know what the function of these proteins were, either in other animals, such as the MHC proteins, or in humans, but we now know that the function of MHC proteins are to bind peptides to present these T cells to start the adaptive immune system. And then these are cartoons over here. class 1 and class 2, and the peptides are in these binding grooves here, and the majority of the important changes in the alleles occur in those grooves. So having different amino acids in the groove will allow them to bind different peptides. So we know that when we're talking about mice, one way to avoid rejection is to transplant tissue in syngeneic mice from one mouse to another. So it's the same MHC genes, same MHC proteins, two mice can have exchange tissue. So the question is, what's the equivalent in humans? Does anybody know? And then it is, yeah. So in 1954, at the Brigham's Hospital in Boston, the first successful kidney transplant was actually done between these two identical twins here. And this person here, this is Joseph Murray, whose picture I showed the last time. He's a plastic surgeon who was in Britain in World War II, and he was the one that was interested in trying to get skin grafts to work on burn patients. And this kind of triggered his interest in why those skin grafts were rejected, and that led to his research into transplantation when he went back to Boston, and so he was part of the first successful team. So one of the experiments that they did on these twins, before they transplanted the kidney, the recipient had glomerulonephritis and end-stage kidney disease, and so he would have died without, and they didn't have dialysis at that point, he would have died. But one of the things that they did, just like in mice, is they took skin grafts from the donor and they put them on the patient who's going to be the recipient, and they waited, and the skin graft did not reject. So they thought, well, that means that, they are identical enough that if we were to try with something more radical like the kidney, then it wouldn't be rejected. And because we have two kidneys, you can actually take the kidney from a living donor, and the donor can survive with one kidney, and then the recipient who has end-stage kidney disease can then get a kidney. So do you have any idea which of these two is the recipient and which is the donor? So this is the recipient, and he's smiling because now he has a functioning kidney, and he's not uremic, and so he feels pretty good. This is the donor, and he's frowning. Well, the reason he's frowning is not because he just donated his kidney, but he hurts, he has pain, and he's kind of splinting to one side, to his left side. And that's because, anatomically, where's the kidney? It's under the 12th rib. So the original kidney procurement operation required making a flank incision, and they do this if you have kidney cancer, and they need to do a radical nephrectomy. But they actually remove part of the 12th kidney, rib, to get at the kidney, and then they take the kidney out. And so he has kind of a big incision, and he's kind of still hurting, recovering from that operation. Whereas the recipient, we know the kidney goes where? So it's down in the iliac fossa, so it's an abdominal surgery, but I mean, it's not quite the same as going through the major flank muscles. And this donor lived 30 or 40 years after. Unfortunately, the recipient had recurrent glomerulonephritis, and he lasted, a couple of years, at least. And they didn't really need any anti-rejection medication because they were identical. And they didn't really have a lot of good immunosuppression at that point to use. And the donor ended up marrying the ICU nurse who was taking care of him. So, other than identical twins, how do you determine what could be a compatible organ for an organ recipient? So I have this case. So this is kind of hypothetical what Susie's HLA makeup might have been if we had typed her. So we only have three here, two class 1 A and B, and then class 2 DRB. But this is her makeup, so she has one protein or one gene from her father, one from her mother. So she has 2 A's, 2 B's, and 2 DRBs. And so in this case, we typed all of her siblings. So which one do you think might be a match? C. Sibling number 3. So can we just go ahead and transplant at this point? Or is it we still don't quite know what to do? So that's the answer, and we'll show you why. So I showed this the other time, last time, and part of the problem now is we know that there are a very large number of different genes that code for different alleles, but I said that they're not all that important because a lot of them, substitutions, in the genetic code substitutions really just make synonymous proteins in the end. And so these are the kind of number of proteins that we have here, and not all of them are expressed on the surface where they might be reactive, they might be exposed to recipient immune system. We talked about why it's so polymorphic. So one thing that makes it easier is what's actually called haplotype inheritance. Or what is that? So generally when we're talking about your genetic makeup, we're talking about your genotype, and usually you think all of your genes are just kind of inherited randomly from your parents, right? I mean, you have siblings, you might not all have the same eye color, hair color, so on and so on. You might have different disease susceptibility genes. You might have a sibling who has asthma, you don't, that kind of thing. But when certain genes are close together or for some other reason, they could be linked, and so they're actually not inherited completely independently of each other, but they're inherited in what's called a haplotype, or a group of genes. And so that's true with the MHC and the HLA system. So this would be the father's HLA haplotype here. He has 2, and the mother has 2, and the chromosomes segregate separately. So how many different combinations of them can there be? 4, right? So if this is Susie's inheritance, she's inherited this chromosome, or this haplotype from the father, and then this one from the mother. And so these are the other combinations of those separations of those haplotypes. So you have a 1 in 4 chance of matching with somebody else. So in this group of siblings, who is she going to match with? So again, it's sibling number 3 down here. So sibling number 3 happen to have the same haplotype inheritance from the parents as Susie, and for these 3 particular genes, they're an identical match. And so she might be a very good donor for Susie. So this is one situation where having more siblings, if you need an organ, is really helpful because then you're more likely to have a match. And this is going to come up later with bone marrow transplants or stem cell transplants as well. So one of my sisters had acute lymphoblastic leukemia, needed a transplant. I had already been typed because I typed myself in the HLA laboratory, so I didn't match her. And then my younger sister was typed, and she was a complete match for my sister with the leukemia. And it turns out that the haplotype that I had from my father and mother, presuming we had the same father, is completely different than either of my 2 sisters. And so why is matching important? So for the purposes of this diagram, we only have A, B, and DR. Again, but you can see, so the 0 mismatch is this top line, And you can see at 1 year, everybody's pretty good. So transplant centers are ranked on how well their organs, especially kidneys, survive in 1 year. And you should, if you're good, you should have pretty close to 100% survival at 1 year. But you see that even when you have a complete match, there's a fall off. But if you're complete match here compared with a complete mismatch at all three of these, that there's a difference between about 75% and 65%. So a 10% difference when you're not completely matched at all. And partial matching is kind of somewhere in between. So what we're looking for is, the best match possible. So we want to talk a little bit about transplant rejection, come back to why that matching is going to be important. And so we know that there are basically three types of rejection: hyperacute rejection, acute rejection, and chronic rejection. And hyperacute rejection comes about because you have pre-existing antibody in the recipient that is already able to react with donor tissue and reject it. And cellular develops if there's a mismatch in the HLA antigens between the recipient and the donor. And then if there's that mismatch, then over time there can be what's called chronic rejection. So it is hyperacute rejection. As I said, it's pre-existing antibody. And so what happens, and what's going to be important is we're going to find out is that you have ABO antigens on your endothelium. So ABO antigens are not just on red blood cells, although it's very important for transfusions, because if you have ABO antibody and you get the wrong red blood cell type, you're going to get immediate rejection within red blood cells. Means you're going to lyse the red blood cells, release a lot of hemoglobin, which we know is toxic to the kidney tubules, and you end up with AKI and ATM. So we try to match red blood cells, but it also turns out that you have both ABO antigens and your HLA antigens on the endothelium. And if for some reason the recipient already has antibody, and we know from transfusions that people who are of a different type already have circulating anti-red cell antibody that's not of their own type. So they either have ABO, anti-ABO antibody or anti-HLA antibody that's circulating, and then it binds complement, and you can activate clotting cascade, get inflammation, thrombosis, and eventually, this is a mouse heart model. You transplant between non-syndemic mice. Right away, the heart looks pretty good. Blood starts to circulate, and then it clots off, and the muscle myocardium immediately becomes ischemic and infarcted, and it looks like this. And unfortunately, there have been rare cases of hyperacute rejection in human heart transplants. A number of years ago, there was a young woman recipient, I think at Duke, and there was some mix-up in communication, and she actually got a heart that was pretty good for HLA typing, but it turned out to be the wrong blood type. And she had a hyperacute rejection of her transplanted heart, but they were able to support her on cardiac bypass for a while until they got a second heart for her, and so she was okay. But there's a lot of concern with transplant centers to make sure that both HLA matching is compatible and that they have the right ABO type. And hyperacute rejection in the kidney can look like this. So here are microthrombi in the glomerular capillaries, and that's really going to gum up the circulation in the kidney. So when we come back to what we want the laboratory to do, we want to type the recipient, we want to type the recipients first. And so we have a wait list for patients who need organs. and when they're at their recipient transplant center, is they're all typed when they come in, and then their typing goes into a computer. And so for now, for all solid organs, we type for class 1 A, B, and C, and also class 2 D, R, and DP. So an ideal match now would be 10 out of 10, more than 6 out of 6. And then we also type the recipient and the donor's ABO typing. And so some recipients can have existing HLA antibodies, and we know that from the way we originally found the HLA types is that women who've had prior pregnancies often will have HLA antibodies against the father's haplotype that the neonate has inherited from the father. And so they already have HLA antibody. And so if they're a kidney recipient, we need to know what those antibodies are so that any potential donor for them does not have a type against which they already have antibodies, which is also why it's really hard for children to donate solid organ kidneys to their parents, to their mothers especially. That makes sense? And then before we started...
Just a quick question. So with class 1, for example, we get some of those genes from our mom, some from our dad. Are both of those expressed?
Yeah. So they're all of the HLA genes are co-dominant.
Okay.
So that's why you have 2 of everything that you're inheriting.
And so that's where the 10 out of 10 comes from?
Right.
Okay.
And then you can also have pre-existing HLA antibody from blood transfusions. It's not so much of a problem now because when you donate, the red blood cells are leuko-reduced with filters, so they have many fewer white blood cells in them than they did before. And this is important for transfusion reactions, but it's also important for preventing the development of HLA antibodies. And so prior to the use of epogen to stimulate red blood cell production in kidney patients with end-stage kidney disease, a lot of times the patients who were potential recipients also had a lot of HLA antibody because the blood transfusions that they got to maintain their hemoglobin level also gave them a lot of lymphocytes that have HLA antigens on them, so they develop antibody against those. And then we also have the situation now where many kidney patients have had one transplant and then it eventually develops chronic rejection, stops working, and so they may have antibody from their first transplant that has to be taken into consideration for their second one. So we did blood typing on the father and the mother. And so again, here's Susie, and you can see the distribution of blood types. So in this case, A and B are dominant over O. O is basically the lack of any antigen. So if you're type B, you don't know if you're BB or BO, and so on. But in your children, you could tell if the other spouse is different. And sometimes there can be confusion about, well, I'm not really the parent because my kids are this blood type, but I mean, it could be perfectly okay. And before we had better ways of identifying proper offspring or proper parentage, red blood cell typing was one of the early methods of determining parentage. And that's really only important because in the legal system, if a mother is only trying to support herself with a child, the legal system will often go after the father to pay child support. And our laboratory at St. Francis used to do some parentage testing for that. So anyway, the same thing. Here, they both match. So again, it turns out that Susie blood type O We also tested her for pre-existing HLA antibodies. She doesn't have it. Sibling number 3 is a good match, is also type O, so the donor to use is sister number 3.
What if there wasn't a perfect match?
Huh?
What if there wasn't a perfect match? Who's the best?
So you can still transplant. So that's why I showed the first graph, showed how many were the complete match, and we still transplant even if there's a complete mismatch. But the longevity of that graph goes down. So it's really good if you can get a complete match. So what makes her a successful kidney transplant is they're ABO compatible, no donor-specific antibody in the recipient, and then matching. But the matching, so to answer your question, is not the most important, but to have 0 antigen mismatch or a complete match is really the best. And so when patients are in the computer trying to match nationally between a kidney that comes up for donation, they will look through the whole national list, and if there's a recipient in Maine who is a complete match for a donor from Hawaii, that organ, that kidney will go there for that recipient, because that gives that recipient the best chance of not developing any new HLA antibody.
Sorry, is there any ranking system of matching HLA-A is more important or matching HLA DRB is more important? Is there any ranking system of the importance of them, or are they all about equal?
So generally for solid organs, they're about the same. The differences that might come up is that, even though we talk about generating HLA antibody or HLA T, reactive T cells, is some, I think DP, for example, tends to be more expressed and maybe is, but don't quote me on that, or is slightly more antigenic for some reason. So, but overall, the differences in survival and rejection and for solid organs is really not any different between the different classes. Bone marrow transplant, different story. So there are a lot of non-HLA matching factors that are important. So for kidney, because it's going not back in the regular place, it doesn't really make any difference whether it's bigger or a little bit smaller than the person's native kidneys. It might make a difference for pediatric patients. So pediatric patients will tend to get kidneys from smaller individuals or from another pediatric donor. Also because you do need a certain amount of glomerular mass to function properly. Sometimes, if there's no other donor available, 2 pediatric kidneys will go into 1 adult recipient. But it's very important for heart and liver, because the liver is going to go back where the original liver is, and obviously the heart is going to go back. So it's really kind of hard to put a heart from a burly, brain-dead motorcyclist into a small, middle-aged woman. So sometimes that's why it's difficult to get organs. And then it makes a difference in terms of donor source, whether the donor is living or deceased, because generally living donors have a decreased ischemic time. You take it out in one room and then you turn it over and you put it right into the recipient. And so here, ischemia time is important. The longer a donor organ is out of the body, the less likely it is to function at 100%. And then anti-rejection regimens, a little bit different for different organs, and that can make a difference in the successful outcome. So generally in Hawaii, we're kind of at a disadvantage because we're already 2,500 miles from the West Coast, 5,000 miles from the East Coast, and we haven't really been able to donate a lot of lungs from our local deceased donors. The first case we did turned out to be where there was a transplant lung surgeon from UCSF who was here for a meeting, and they were talking to our local procurement group, which is where I got this T-shirt. I currently sit on the medical advisory committee, but they were talking about, well, we'd really like to be able to coordinate timing with at least centers on the West Coast to maybe be able to get lungs to those centers to be able to transplant them. And then right around the time they were talking, and they had a recipient who really had good lungs that would have been good for as a donor, and so the surgeon actually stayed, and they had a recipient at UCF who would use those lungs. So he actually stayed, and he helped procure the lungs here, and he flew back with them. And they were able to cut down on the total time that he was traveling and get the lungs into the recipient pretty quickly. And this also indirectly has helped local recipients, because there was a Big Island recipient who was waiting in San Francisco who received another pair of lungs from a local donor a little bit later on. And he's back on the Big Island, he's doing well. So in this case, they're able to get the lungs all the way to the East Coast. There's a 50-year-old donor. The ischemia time was 24 hours, but he had good lung function, and the recipient is doing well. And so one of the things that's come up with technology is all the organs are usually just transported cold. So you take out the kidney or the liver, you put it in a plastic bag, pack it in ice, put it in a cooler, and you take it on the plane, and you hope you don't forget it somewhere. But now we have all of these powered perfusion devices where we can run fluid through the organs. We can actually pump them, and the lung device has gotten so good that you can actually take a kind of a bad lung and make it better. You can even treat minor infections by running antibiotics through it, and Toronto has been a pioneer in this kind of perfusion. And then there's these different devices for heart, liver, and then recently Legacy of Life, which is a local procurement system here, has been trying this device for kidneys, and it works very well. The problem is it's kind of expensive, and the other problem is you have to have one here that's ready to take the kidneys. If it's going to somewhere on the continent, it gets there, and then how do you get the machine back? So a lot of the barriers to transplantation are really not the biologic ones, but they're the way the system works, and they're financial, because all of this costs money, and we really don't have enough. So to complete the testing, test the red blood cells, and then finally, when you get a donor that you want, again, you want to make sure that they're the same ABO type, and then you take donor lymphocytes and you do a tissue cross-match, similar to a red blood cell cross-match, where you can take donor lymphocytes, and even if you think the recipient doesn't have any antibody, you mix them together and you see whether the lymphocytes clump up or die, or whether they don't and they're compatible. So how do you prevent hyperacute rejection? Well, you do the red cell cross-match. So even when there's incompatible ABO types, the rejection rate is only about 25%. I mean, you would think it would be higher, but you kind of don't want to take that risk. And then you do the tissue cross-match, and if there's a positive cross-match, there's an 80% chance of rejection. If you have a negative cross-match, it's not completely sensitive, so you still have a 4% chance of having hyperacute rejection. So I showed this the last time, and this is the usual situation of the adaptive immune system, where we have what's called MHC restriction, where technically your own T cells can only recognize your own HLA from the antigen-presenting cells. So if there's a different antigen-presenting cell makeup, then your own T cells don't recognize it. And the exception to that is in a situation where a different donor antigen-presenting cell are presenting HLA peptides that can be identified by recipient T cell receptors. And I think at this point we still don't completely understand why that is, but as I said the last time, we seem to have a fairly large number of our T cells that can recognize HLA peptides on somebody else's antigen-presenting cells, compared with the way we can recognize peptides from pathogens in our own antigen-presenting cells. So there are two different pathways for acute rejection. It's so-called direct pathway, and this is where you have donor antigen-presenting cells that are presenting donor HLA peptides to recipient CD4 or CD8 cells that kick off the cellular response. So you can get recipient CD8 cells to become cytotoxic, and then they can come in and attack the recipient kidney, or you get CD4 help cells and make interferon, kick off macrophage activation. So the T cell response, final cellular rejection is a type 4 rejection, macrophages and cytotoxic T cells, and so on. What's interesting recently is what's called a semi-direct pathway, and it's actually thought that this is what's been going on all along. So the direct pathway is not necessarily donor dendritic cells in the graft, because in experimental models, you can flush out all of the donor antigen presenting cells and still get a direct rejection response. So what we think is going on is that donor dendritic cells make exosomes, and the exosomes are composed of the HLA complex of the donor with the donor HLA peptides in them, and then those exosomes fuse with the recipient dendritic cells. So the recipient dendritic cells are now expressing donor HLA molecules or with donor HLA peptides to activate these CD4 and CD8 cells. But the main thing to remember is that the interaction is between the donor HLA presenting molecules with HLA peptide to recipient T cell receptors. Did everybody get that? So again, it's like this: it's a donor MHC or HLA presenting donor HLA against the recipient T cell. The indirect pathway is once the kidney or the organ is in the body, what happens is they shed a lot of HLA in proteins from the surface, other proteins as well, and that's because there's a lot of ischemic damage, there's reperfusion injury, the antigens float out into the blood, and then they're picked up by recipient antigen-presenting cells, which break them down and present, again, donor HLA peptides to recipient CD4 or CD8 cells. And then they also can be picked up by the B cell receptors on recipient B cells, and then with the help from CD4 cells, you're eventually going to make anti-HLA donor antibody. So acute cellular rejection is shown here. You can see infiltration by lymphocytes into the tubules of the kidney, causing tubular damage. Here's another one down here. Antibody-mediated rejection, a little bit different. So if you have anti-donor HLA antibodies, they can come in, they bind into, again, like hyperacute rejection, they bind to the endothelium, and then they can activate adherent molecules, which then allow macrophages and neutrophils from the circulation to come out into the interstitium and then cause acute inflammation. So in this picture here, you see margination of the neutrophils in this peritubular capillary that has anti-HLA antibody bound to it. And the antibody, unlike when you have glomerulonephritis and you have antibody to glomerulus, is really hard to pick up by immunofluorescence. But because it's also capturing complement and fragments of the complement pathway, one of which is C4D, are deposited in these areas where the antibody and the acute inflammation is going on, and you can stain for C4D in kidney biopsies. So this is the immunofluorescence stain here. This is an immunostain here showing the deposition of C4D complement component, indicating that there's antibody rejection going on here. So this is a different case. The recipient has this makeup. Again, 2A1, 2 A's, A1, A66, 2 B's, 2 C's, and then the donor has this HLA makeup. So 1, 3, and then this. So how many alleles are different? 4, right? So this is a four antigen mismatch. So they match here. 1, this is different. This is different. This is different. So that's 3. They both have C6, but this is different. So there are 4 antigen mismatch. The recipient did not have any HLA antibody prior, and so it looked like his donor, not a really good match, but a match. And the patient did well until about 2 weeks later when his creatinine started to go up and he wasn't feeling very well. So what's going on? Does he have hyperacute rejection, acute cellular rejection, antibody rejection, something else? What do you think? So it could be that he's rejecting and he has an acute cellular T-cell mediated rejection or antibody mediated rejection, or just be something else going on in the kidney. He could have some kind of infection, maybe. So how are you going to work it out? Well, what test do you want to do? Based on what test we know we can do. So we can look for more anti-HLA antibody. We know that pre-transplant you had no antibody, and now we can look for antibody and see what that antibody is. And so this is what's called donor-specific or DSA antibody. And what we found in him is that he's now developed an anti-A3, B62, and C7. So these 3 out of the 4 that he was mismatched with. And so this could either be acute cellular or antibody-mediated rejection.
Just to clarify, when you say acute cellular versus acute antibody mediated, is that the direct versus indirect pathways?
It's either. So a final pathway is the development of CD8 cytolytic, cytotoxic T cells, which would be involved in the cellular rejection mode, right? Because they come in and they can kill the kidney cells. Or, once you get the T cells activated, turning into plasma, the plasma cells can make the HLA antibody, which can then come in and attack the kidney. So again, the same as in hyperacute rejection antibody-mediated human rejection is donor-specific antibody adhering to HLA antigens on the endothelium, causing thrombosis. And endotheliitis, neutrophils marginate, come out, cause inflammation. So one of the tests we can do in the lab is to detect new donor HLA-specific antibodies. It looks skeptical. And I'm showing this graph again. So what happens over time is even though you have the best chance of survival with a complete match or zero mismatch, that they all kind of have the same slope. So at 12 years, the survival is much less than it was at 1 year. And the reason this is because even though we have pretty good immunosuppression short term, there's still a lot of kind of chronic rejection going on in the background that we either can't identify in any way or treat more immediately, and so we have chronic rejection. Particularly if you develop a lot of donor-specific antibody, your risks for chronic rejection are much greater. And then so with this ongoing inflammation, you get a lot of cytokine release. Again, you get a lot of continued response of antigen-presenting cells to present antigen, and then they come in, and you get transition from type 1 to type 2 macrophages. And type 2 macrophages, although they help in the healing process, the biggest problem is they make TGF-beta, which causes fibroblasts to proliferate, and you get fibrosis. So you get intimal hyperplasia here. We can see this in this kidney arteriole here, and you get a lot of fibrosis in the surrounding areas. And you eventually get an end-stage kidney with a lot of fibrotic glomeruli. So how do we prevent rejection? I'm sure you present everyone had this in their learning, one of their learning issues. So there are a variety of pathways, and so the idea is to kind of try to interrupt the interaction of the antigen-presenting cell with the recipient T cell and the T cell receptor. And so immediately what you can do is there are drugs that can attack the T cell receptor so that it can't really interact with the antigen-presenting cell. Here's this HLA peptide in here. The other thing we know is that to actually get the T cells to respond, you need to have co-stimulation. So one of the co-stimulating molecules is recipient of CD28 interacting with B7. So there's a monoclonal antibody against that's similar to CTLA-4. So CTLA-4 is part of the adaptive immune system that actually comes in and shuts down the immune system after a pathogen has been eliminated. And so what you can, and it does that by interacting with the co-stimulatory receptor here and the ligand here. And so we have this monoclonal antibody that acts like CTLA-4, comes in, blocks co-stimulation, and then shuts down activation of the T cell. If you don't have that, co-stimulation eventually results in IL-2 production. And IL-2 is a growth factor for T cell. So we know that in the normal response, IL-2 acts here on the receptor, and then it works through this pathway here. So sirolimus interacts with the FK binding protein, and it interferes with this transcription factor here so that you don't get proliferation and development of IL-2. And then similarly, tacrolimus, which is the newer calcineurin inhibitor, works here. So once the T cell receptor is activated, then it works through this pathway and has a transition factor that it's going to make IL-2. Tacrolimus interferes with this pathway. So you shut down IL-2 production to prevent proliferation and activation of the T cells. And then we have these more non-specific drugs which interfere with actual DNA synthesis. And what's important is that unlike other cells in the body, lymphocytes do not have a salvage pathway for purine synthesis. So they will actually preferentially cause the destruction or the inhibition of the proliferation of T cells, which is why they're important in autoimmune diseases, immunosuppression for anti-rejection, and then also a lot of lymphoid malignancies. And then the other way we have now of trying to cut down on rejection is here showing the donor HLA protein interacting with the B cell receptor, and then stimulation by CD4 cells eventually makes this plasma cell that makes anti-donor HLA antibody. We have rituximab, which targets CD20 on B cells. It's used a lot for B cell leukemias or lymphomas, but we're now using it in transplant to kind of kill off the B cells that might make anti-HLA antibody. The T cell drugs that we have can also work on these recipient CD4 cells, the follicular helper cells that are coming in to help B cells become plasma cells. And then we have drugs for multiple myeloma, which is a plasma cell malignancy, are also being used to kill off some of the plasma cells and making anti-donor HLA antibody. And then if that doesn't work, sometimes we have used plasmapheresis to try to remove more immediately some of the donor anti-HLA antibody, or we use a lot of IVIG too, which binds to antibody non-specifically. So what are some of the non-transplant or non-rejection complications? A lot of complications from the drugs. So corticosteroids cause osteoporosis. They give you moon phases, vascular necrosis of the femoral heads, cyclosporin, which we don't really use anymore. The oldest original calcineurin inhibitor caused a lot of hirsutism, causes hypertension, and then actually is nephrotoxic. So it's really kind of paradoxical that we're using a drug to kind of keep the kidney from going bad that really helps the kidney go bad. And there have actually been cases when these drugs are used for liver or heart transplants where the recipients then have to get a kidney transplant because they had cyclosporin nephropathy. And then the tacrolimus also is nephrotoxic. And then one of the other side effects is tremor. So I told my tutorial group that a few years ago there was a well-known transplant surgeon at NYU who had a congenital cardiac defect. He eventually did have a heart transplant. He elected not to take tacrolimus because he wanted to operate. And at that point, a few years ago, sirolimus was a little more in clinical trials for anti-rejection because tacrolimus was more kind of the standard drug. But he went on sirolimus and has done very well. There have been a number of infections, both related to the donor organ. So, some of the herpes viruses, CMV, HIV has been transmitted that way, West Nile virus. So sometimes what happens is patients who are brain dead make good deceased donor organs, but sometimes we don't have a good history on why they have brain death. So West Nile virus is a neurotropic virus, rabies is neurotropic, CJD is neurotropic, so they've gotten that. And then with recipient immunosuppression, if you're CMV positive, you can get reactivation of CMV, so we have CMV prophylaxis. We also have this other virus called BK that can attack the kidney, and then again another herpes virus, EBV. And then you get fungal infections, and then also you can get malignancies. So there have been cases of transmitted things like renal cell carcinoma in the donor organ, and then immunosuppression causes to be more susceptible. So in Hawaii, we have a lot of patients who get squamous cell carcinomas. So this kind of just shows the flow of a deceased organ flow. Our OPO here is called Legacy of Life. And so they work up all the deceased donors, make sure they're suitable. They look to see if that donor has given permission to view their driver's license or some other registry. And now, even if the family doesn't want that donor to be used, state law says that we can use that donor for organs. And then they coordinate procurement with the national organization. So the HLA laboratory is up at the Weinberg building in St. Francis. They do all the typing on the recipients on the wait list. They do all the donor typing. They do all the infectious disease markers the same way we look at blood transfusions, and they do the final cost match. And then the final organization that does the matching is on the continent. And Queens is our local transplant center that does the procurement of the donors and then transplants into the recipient. And so in 2024, they transplanted 7 kidneys, 63 deceased, 7 living, and they did 23 livers. There was a recent TV thing, I think, on KHON. Anybody see that? It was unfortunately kind of very misleading. So what turned out, it was a recipient on kidney dialysis who wanted to get a kidney from his daughter. It turned out his daughter had proteinuria, so she was not a suitable donor. Even after they made her lose 80 pounds in weight. I mean, these are all issues for what makes us a good donor and to be able to safely take an organ from a living donor. Anyway, he was saying that there haven't been that many transplants in Hawaii, and even though he thinks we have a lot of donors here, which we don't, that most of those organs went to the continent. And they didn't really go into why that is. And so now we're at the legislature all riled up again, looking into, well, why can't we keep more donor organs here? And we wish we could, but I mean, there are a lot of reasons why we can't. So I showed this before, just to kind of remotivate you as to what the issues are. You know, how many people we have waiting here? Almost 300 waiting for kidneys in Hawaii. So if you're an organ donor, you have a legacy that you carry on. And we had a campaign a little while back, people saying they signed up on their driver's license. So any questions?
You're on